So let me just start with this only. Yeah. So, yeah, like I said, chemolithotrophy involves two distinct processes. One is where the electron donor passes on its electron to the electron transport chain, very similar to what you observed in chemoorganotrophy. So basically, it's this chain. And as the electrons are transferred, a proton motive force is generated, which is involved in oxidative phosphorylation. Okay. But like here you saw that the same source was responsible for biosynthesis. In the case of chemolithotrophy, mostly the carbon source is CO2. Matlab, this is autotrophy. Okay. And this is the process which is distinct. So because the sources are different, here the ATP generated from the electron transport chain or from oxidative phosphorylation is used up together with NADPH. Biosynthesis may, if you remember, NADPH was being used, and there is biosynthesis. Okay. So, like I said, the focus for us is energy generation, but I'll briefly also cover how carbon dioxide is fixed in this case. Uh, Now, there can be a variety of electron donors, with the most common substance being hydrogen. Okay. And then there could be sulfur compounds, there could also be nitrogen compounds, and as you can see, ferrous ion as well, that can serve as a donor for the electron. The electron acceptor is mostly oxygen. Mostly that's a very common acceptor. We will talk about two cases. We'll talk about hydrogen and nitrogen compounds. Hydrogen because it happens to be quite energy efficient, and nitrogen because we've covered something just the opposite of it in chemo anaerobic respiration. Now, before I go into these two, um, I have to show you this. This is a redox tower, by which you can understand that the amount of ATP generated in the process will vary depending on the donor used. Okay, here I'm showing you the hydrogen couple. The energy yield in chemolithotrophy it will be less than aerobic respiration, since the largest delta E0 prime, if you see, is found using glucose as an electron donor and oxygen as an electron acceptor. But how much less than the 32 to 38 molecules of ATP, that depends on the actual donor and the acceptor being used. You can understand, the smaller the difference between the donor and the acceptor, the lesser ATP will be formed. One more thing that you have to note here in the redox tower is that if a chemolithoautotroph is using an electron donor with a higher redox potential, somewhere here, then please note NAD plus NADH here. If it is higher than the value than the redox potential for NAD plus NADH, then the lithotroph must actually push the electrons back up the redox tower to synthesize NADH. This is not going to be a favorable process. So you will need ATP to actually push it to make the reducing power. This is called as reverse electron flow, which you haven't come across till now. This is special to chemolithotropy, and also later you will see in the next lecture also. Now, the energy from the PMF, which is consumed to drive electrons in the reverse direction. So, the same energy which is used to synthesize ATP, a part of it is then deviated for formation of NADH by pushing it against the only exception, if you see here, the only electron donor which has a more negative redox potential happens to be when hydrogen is the electron donor. So only in this case there will be no reverse electron flow. For all the other electron donors, you need to invest energy in forming NADH as well. Now coming to hydrogen oxidizing bacteria, these oxidize hydrogen gas with the use of an enzyme called as hydrogenase. And you have both aerobic and anaerobic hydrogen oxidizers. So the aerobic one would use oxygen as electron acceptor, while the anaerobic one would use sulfate or nitrogen dioxide as electron acceptor. Most that require oxygen as electron acceptor, they also grow best under microaerophilic condition, as the enzyme, it is about the sensitivity of the enzyme. This is inhibited by the presence of oxygen. So mostly it is microaerophiles which would prefer nitrogen as the electron donor. Now, this ability to use both organic and inorganic substrates makes them successful in a number of niches. So you will actually find them in some of the extremophilic conditions as well. And there are two types of hydrogenases which have been identified. There's one which is a membrane-bound hydrogenase, while there's another one which is a cytoplasmic hydrogenase. And इससे तुम्हें अब समझ में आ रहा है कि where it is located in the redox tower. Yeah. So I was talking about a membrane integrated hydrogenase and a cytoplasmic hydrogenase. So hydrogen is oxidized by the membrane bound. So here, if you see the membrane bound hydrogenase, causing a protein pump. So here you see this entering into an electron transport chain, which functions very similar to what you saw in oxidative phosphorylation earlier in respiration, जहाँ पे कि protons are pumped out and then the enzyme ATP synthase would synthesize ATP with the PMF that is generated. And the terminal electron acceptor, like I said, is mostly oxygen. So here you see oxygen taking up the electron forming water. Now, in many organisms, a second hydrogenase is found, which is the cytoplasmic hydrogenase, which is in fact used to generate the reducing power, okay, in the form of NADH. And it is this NADH which then helps, together with ATP, helps in the fixation of carbon dioxide. This is through a process called as Kelvin-Benson cycle, but I will not go into the detail of Kelvin-Benson cycle. Fixation is not something that is going to be discussed in this course. ये समझ में आया कि हाइड्रोजन redox tower में कहाँ situated है, and so the primary hydrogenase is membrane bound, which passes on the electron to the electron transport chain ATP synthesis के लिए. In some organism there is this extra hydrogenase which serves as NADH, which functions in synthesis of NADH. Now, the other electron donor that I'll talk about is going to be nitrogenous. And basically why? Because we have talked about, if you remember, we have talked about a method wherein nitrogen was being lost to the environment. So nitrates and nitrite and other oxides, and ultimately it was forming nitrogen. किसी को याद है हम लोग ने ये कहाँ किया था? ये प्रोसेस डीनाइट्रिफिकेशन का किस मेटाबॉलिक पाथवे में हम लोग ने देखा था? Anybody? अभी तक we have covered aerobic respiration, anaerobic respiration and fermentation. Where have we come across denitrification, students?
Ma'am, in anaerobic respiration.
Correct. In anaerobic respiration, right? So you saw that instead of oxygen, it was the nitrogen moieties which were the terminal electron acceptors, and ultimately I told you the positives and negatives of the process as well. For example, in agriculture, this will be a major loss. Something fixed as nitrate is being lost as nitrogen gas, and an inert moiety which cannot be taken up by plants. But this is a process which was helpful, for example, in bringing down the algal blooms or increasing the potability of water. So this is what we have talked about. Hence, I took up the next process, which is actually a part of the nitrogen cycle for chemolithotropy. So basically, nitrogen oxidizers, मतलब when these are used as a source of electron, यहाँ पे ये source of electron है, और denitrification में difference ये था कि वहाँ वो acceptors थे. Nitrogen moieties were actually acceptors. Okay. So here there are two steps which make up nitrification. One is conversion of ammonium to nitrite, and the next step is nitrite to nitrate. Okay, oxidation हो रहा है, वहाँ reduction हो रहा था, accept कर रहा था electron तो. So these are carried out by two distinct genera. Nitrosomonas is the genera which is involved in converting ammonium to nitrite, while Nitrobacter are species which convert nitrite to nitrate. And of course इस केस में भी कार्बन डाइऑक्साइड फिक्सेशन कैल्विन साइकिल से ही होता है, हाँ. लेकिन here again the concept that I was trying to explain to you, look at where the nitrate nitrite couple is. So here it is, for it to form NADH, electrons have to be pushed using ATP, and then NADH can be generated. NADH and NADPH, because NADPH, as I mentioned to you, is the one involved in biosynthesis. So here, this is the reason why I'm showing you the redox tower every time. So this is the first process, nitrification, ammonium to nitrite, wherein there are two enzymes involved. One is an ammonia monooxygenase. It's actually a two-step process by which ammonia is first converted to hydroxylamine, and then hydroxylamine is converted to nitrite. So this is what I'm trying to say, to show you here, that look, this is the electron transport chain. This is the normal trend. Okay, so it passes on, and in the process throws off or generates a proton motive force. Achha, students, this is a picture that I've taken from I think Brock. So that is why you see this kind of disfigured thing. Anyway, so but again, when the cell requires NADH, it has to push it from this electron. This is quinol, a lipid soluble electron carrier. It has to the quinol has to actually use ATP to take it up or pass it on to a more negative a moiety with a more negative redox potential, that is NAD plus, and that's how NADH is generated. And this is exactly the same case with nitrite to nitrate conversion also, that there has to be, the enzyme is NXR, nitrite oxide reductase. But here also there is a reverse electron flow which has to happen. And so a part of energy is actually, hence the process is not energy efficient, as you can understand, if it has to actually distract some of its energy for NADH formation. Yeah, so this is the case in both the steps of nitrification, whether it is ammonia to nitrite or nitrite to nitrate. Yeah, so what we talked about right now was this, the electron transport chain. And I said this module is concentrating only on ATP generation. But just to very briefly tell you what happens to the carbon dioxide, what happens to the NADPH and ATP that is generated, it enters into, like I said, a cycle called as Calvin cycle. And this cycle is also important in the next topic, which is phototrophy, wherein CO2 is fixed. So just to give an idea, I'm not showing you the moieties or the enzymes. Just to give you an idea, if you start off with 3CO2, and why usually you start off with 3CO2, because the simplest sugar that you can think of is a phosphoglycerate, PGA, जो तुमने पहले देखा होगा, which can form. So in certain books you will see 6CO2 being fixed so that you can come up with a glucose. And here what happens is, there are three molecules. So 3CO2 matlab three molecules of the of a compound called as rubisco. I'll tell you what. Maybe I'll start off here. So RUBP, which is ribulose bisphosphate. Bisphosphate, keto, 5 carbon, नाम से तुम्हें समझ में आ रहा है, हाँ? The enzyme rubisco, which is a very common protein, it fixes carbon dioxide, and this is the acceptor of carbon dioxide. So you will see once it's a bis. From your earlier lectures, you know that this is going to be a very unstable molecule. So the CO2 comes and hits this ribulose bisphosphate, such that it then breaks down to 2,3-phosphoglycerate. ऐसा ही कुछ तुमने EMP pathway में देखा था, जहाँ पे कि fructose bisphosphate का breakdown हो रहा था. यहाँ पे CO2 comes and attaches, and you have 2 3 carbon compounds, which then enters into, so it uses up ATP, it uses up NADPH, which has been generated in the phase that we just saw, and then ultimately one glucose comes out, depending on how many carbon you start off from, and then RUBP is regenerated. Okay. So let's really know, this is just to give you an idea of what actually happens. I don't really want you to memorize things. The enzyme is called this is quite a critical enzyme, Rubisco. Rubisco is actually ribulose 1,5 bisphosphate carboxylase, which you just saw. It was carboxylating RUBP, and it's also an oxygenase. And why I'm mentioning this, because like I said, it is the most abundant protein on the earth, and an estimate says that the mass would be like approximately 0.7 gigaton on earth. So this is a process which you will also encounter in phototrophy, but I will stop here. I mean, to be taken up in any further detail. Okay. These are just some interesting papers that I came across, so I thought I would be share. I can those who are interested can go through this. But now tell me if there are any questions from chemolithotrophy, because then I'll move on to phototrophy. And yeah, and it has this second phase which is common. So Calvin-Benson cycle is something which is common. Students, are there any questions from chemolithotrophy? You saw what, you understand what is reverse electron flow, you understand why hydrogen oxidizing bacteria would be more efficient. So for example, a bacteria, if they do have an option to go uh, for a process where an NADH is not required by this process, by the reverse electron flow, that will be beneficial for them, right? Here they are investing a part of the formation of NADH here itself. So it is extracting, or it is compromising with their energy efficiency. राइट सो हाँ पूछो, ये समझ में आ गया कि केमोलिथोट्रॉफी कैसे डिफरेंट है केमो ऑर्गेनोट्रॉफी से। और मुझे नहीं चाहिए कि आप लोग को ये पूरा चेन याद हो कि पहले BC1 CA थी, ये सब मुझे नहीं चाहिए। आपको बस बेसिक कॉन्सेप्ट पता होना चाहिए कि क्या हम आपको यहाँ पे रिवर्स इलेक्ट्रॉन फ्लो, हाइड्रोजन में तो नहीं लेकिन नाइट्रोजन में रिवर्स इलेक्ट्रॉन फ्लो शो कर रहे थे। और यहाँ पे ये कि there are actually two different hydrogenases, one making NADH and the other one involved in electron transport chain. Any questions? Nahi? Then I'll move to phototrophy, which is also going to be a small topic and a related topic. So let me stop sharing this then. तो फोटोसिंथेसिस पता है क्या होता है ना? मुझे बता सकते हो फोटोट्रॉफी और फोटोसिंथेसिस में क्या डिफरेंस हो सकता है? या सिनोनिम है? बोलो, एनीबडी? दीपक जहांगीर? I can't hear you, Deepak. Kashish? Bhavya?
फोटोसिंथेसिस मतलब लाइट के इस्तेमाल से ऑर्गेनिक प्रोडक्ट बनाना। फोटोट्रॉफी कंजम्पशन से रिलेटेड
नहीं। All photosynthetic organisms are phototrophic, but not all phototrophic bacteria, they are photosynthetic. Okay, so photosynthetic organisms are a subset of phototrophic organisms. Okay, next class में जब भी हम मिलेंगे, somebody has a message, ma'am, photosynthesis is a part of, हाँ दीपक, yes, that is what I said. Yes, it's a part of it. Kaise it's a part of it, we will see as we move along. So till now you know of photosynthesis, you've heard of photosynthesis. क्या होता है photosynthesis में? Synthesis by light, right? That is the basic concept कि plants photosynthesize करते हैं. For us, it's not about plants, but about microorganisms. So again back to the nutrition class. तुम लोगों ने ये देखा है कि कैसे the energy source could either be light or it could be chemical compounds. Now to give you a context of where we are, we covered chemoheterotrophs we covered aerobic respiration, anaerobic and fermentation. ये बार-बार हम आपको बता रहे हैं सिर्फ इसलिए बताने के लिए कि where do we stand in the syllabus and in the entire metabolism. And just now we talked about chemoautotroph. ठीक है, ये मैंने कहा था lithotrophs जो हैं वो सारे autotrophs होते हैं, तो synonym इस तरह का आप देखोगे. हाँ, कार्बन डाइऑक्साइड is being used, हाइड्रोजन के to be covered. So when organisms use light, they could be photoautotrophs, wherein carbon dioxide is used, and they could be photoheterotrophs, which use up organic compounds. So hetero referring to organic compounds. And the examples here, photoautotrophs में आपके सारे higher plants आते हैं, and microorganisms में you have certain algae and also some cyanobacteria, and there of course there are some green sulfur, purple sulfur bacteria which are also under the photoautotroph wale category mein. There are very few photoheterotrophs which are termed as or in fact these are the general names, green non-sulfur and purple non-sulfur bacteria. We'll see what these are actually. The requirement for phototrophy which is conversion of light energy, तो basically यहाँ से हम आपको difference बता सकते हैं. आप इनको these are photosynthesizing organisms because they synthesize carbon dioxide से food भी light से ही use होता है. However, heterotrophs they are so prototrophs will include both autotrophs and heterotrophs. However, these are photosynthesizing. Only this subset is photosynthesizing group. So like I said, it's conversion of light energy. So you convert it into, the cells convert it into chemical energy in the form of energy ATP. So यहाँ पे the cell would require a light harvesting machinery. It would require ATP synthesis, which is in line with oxidative phosphorylation. But here what you have is called as photophosphorylation. पहले oxidative phosphorylation था, लेकिन here because the source of generation of ATP is light, the process is called as photophosphorylation. And then there is fixation of CO2. Again, this fixation of CO2 is not something that we are going to really talk about in the course, but we're going to talk about, we will see what the light harvesting machinery is and what is how is ATP synthesized. Now, basically, phototrophic prokaryotes can be oxygenic phototrophic organisms, or they could be anoxygenic phototrophic organisms. Oxygenic में there are plants, लेकिन plants हमारे purview में नहीं है. We are talking about cyanobacteria and oxygenic है, तो you will see कि oxygen release हो रहा है. But anoxygenic में there could be purple bacteria, green bacteria, and heliobacteria. What I want you to see is how they are different in the terms of the light harvesting pigment. So basically, you know, is the light harvesting pigment. This is the one which harvests light. And in the case of bacteria, you have bacteriochlorophylls, and the types that I just mentioned to you, they differ in the kind of pigments that they have. So purple bacteria, they are gram negatives, and they have these pigments localized on the cytoplasmic membrane. Of course, unlike eukaryotes, you don't have chloroplasts here. On the other hand, heliobacteria, they are gram positives, and even here the components are all localized on cytoplasmic membrane. This is a rather recent group, heliobacteria, and they are very unique in that these are the only organisms which have bacteriochlorophyll G as the light harvesting pigment. Now, in this case, the green non-sulfur and the purple non-sulfur are the only photoheterotrophs. जो मैंने इस slide में तुम्हें बताया था कि the non-sulfur bacteria, non-sulfur because they don't use sulfur as the electron donor. So they are the only heterotrophs. All the others are photoautotrophs. And this is an image that I took from Key et al. So they use these three in microbial fuel cells. They were talking, hence this bioelectrochemical system that you see here. Now, in the case of green bacteria, There are special structures. Of course, you cannot categorize them as organelles. जो हम लोग last time जब initial बात कर रहे थे कि because the membrane is not a unit membrane, but it's only in the green bacteria that you come across these structures called as chlorosomes. Okay, and these chlorosomes, the green bacteria are also gram negative, just like purple bacteria. They possess the bacteriochlorophyll, the pigments in this distinct structure. Now, the overall reaction is quite simple. You see here, I mean, this is a simplified overall reaction that I've mentioned. So carbon dioxide is basically, because I said most of them are autotrophs, so carbon dioxide is fixed using light energy. And if it is oxygenic photosynthesis, there is photolysis of water, water splits, and so oxygen is released. However, if it is anoxygenic photosynthesis, it is anything other than water, so you will have no release of oxygen in this case. Coming first to the light harvesting machinery. So for light harvesting, there are pigments, not just chlorophyll, but there are other pigments as well. But the main ones are chlorophylls. Chlorophyll, of course, is found in plants and cyanobacteria. But bacteria would have bacteriochlorophylls. Now, whether it is chlorophyll or bacteriochlorophyll, they both have the similar structure, a distinctive tetrapyrrole ring with magnesium in the center and a long 20 is called a phytone chain. And you can understand this is impregnated onto the membrane. So this lipid moiety actually helps it anchor into the membrane. The differences occur in the substitutions around. So you can see that there are several substitutions which make the pigments different in, so when you talk of bacteriochlorophyll A or B or even C, D, and G, they all differ in just these side chains. But these slight differences, they result in a change in the absorption maxima for these pigments. So if you see that changes, and in fact, this allows several different species of microbes together to optimally utilize the full spectrum. So when each absorbs on a different range of wavelength, they can all have a unique niche without much of a competition. So this is the idea of having different kinds of pigments in different kinds of organisms. So A, B, tumne dekha, C, D in another group of organism, chlorophyll A, B. That makes it utilize the entire spectrum. Now, like I said, there are pigments other than chlorophyll also which are involved in light harvesting. Accessory pigments found in photosynthetic organisms, they actually work in conjunction with the chlorophyll, the main chlorophyll or the bacteriochlorophyll. And the combined action of these chlorophylls and accessory pigments, they again enhance the absorption efficiency across the spectrum. So if you see phycocyanin, phycoerythrin, and carotenoids are the accessory pigments. However, these are the main chlorophylls which I just talked about. So the entire range, again, the absorption efficiency is enhanced here. And how they're arranged is also very interesting. The pigments, whether they are the main pigments, chlorophylls, or the accessory pigments, they are all arranged in the form of light harvesting antenna actually. These are called photocomplexes, and it's shaped like an antenna, or it even functions as an antenna. The photocomplexes, they are organized. There is a central reaction center chlorophyll, basically. It's made up of chlorophylls or bacteriochlorophylls, depending on which organism we are talking about. And about 300 and odd pigments form this kind of a complex. And they are appropriately called antenna, like I said, because they absorb as much light as possible and then funnel it, whether it is the accessory pigment or the main chlorophyll, they actually funnel it all down to this one reaction center. Okay, so the molecules absorb photon, excite an electron to a higher energy level, and then pass on. So through resonance transfer, it actually passes on to this center chlorophyll. When the electrons return to the ground state, it releases another photon with a longer wavelength, and this is how it comes down to the reaction center. A pigment in the photosystem absorbs the appropriate energy level of light, which then boosts its electron to a high energy level. It then passes on. The molecules of chlorophyll and the even carotenoids or phycoerythrins and phycobilins, they are angled such a way so as to ensure that the photons which are released due to fluorescence are directed towards the center. So basically इसीलिए ये structure important है, how they are placed is important. From the reaction center, यहाँ पे सब कुछ funnel हुआ है. So from the reaction center, once there is transfer of energy, this special pair of chlorophyll molecules, they actually then pass on electron to the first electron acceptor. So basically ये पूरा capture कर रहा है light, passing on all this energy to a reaction center, and this then transfers electron to the electron transport chain. In general, the process of phototrophy is same across organisms. So a photosystem antenna, like I said, it absorbs light, funnels the energy to a reaction center. जो हम लोग यहाँ बात कर रहे थे कि सारे pigments they concentrate it to a reaction center. The molecule becomes excited, changing to a more negative reduction potential. So it jumps up the electron tower, and then the electron can be passed on to through an electron transport chain. It can be passed on to various kinds of mediators or transporters of electrons. And in the process, you have generation of proton motive force, जो अभी तक आप देखते आ रहे हो, whether it is chemoorganotrophs or chemolithotrophs. In this process, you have the generation of ATP. Now, here, because the original energy is coming from sunlight, this entire process is called as photophosphorylation, unlike oxidative phosphorylation, which we saw was happening earlier. Now, if the electrons are returned to the special pair of chlorophyll, bacteriochlorophyll, this process is called as cyclic photophosphorylation. Electron is moving back to the same reaction center, and here you can see the process can be repeated over and over again. But if the electrons are diverted elsewhere, for example, in the picture you can see that reduction of NAD plus to NADPH happens from these electrons which are passed on to the electron transport chain. In that case, an external electron source must be used to replenish the system. Okay, otherwise the system is not going to function further. So here, water is being photolyzed to replenish these electrons which are lost from the reaction center chlorophyll. Okay, so this is a common thing which happens both cyclic and non-cyclic, which happens in all cases of phototrophy. Coming specifically to oxygenic phototrophs now, for students who would like to leave at 12, you're free to leave, but I would like to finish this topic here. And those who want to stay can really, I mean, there's not much left, another 10 minutes maybe, but I would not like to leave it midway. Okay, is that fine, students? Deepak Jangir. जिसको जाना है जा सकता है, okay? दीपक, तुम्हारी आवाज को क्या हुआ है? तुम अभी तक तुम्हारा वो ठीक नहीं हुआ. Anyway, ठीक है. So yeah, like I said, all those who wish to leave can leave at 12. Let me check. It's fine, ma'am. ठीक है, दीपक. Yeah. So we'll start with oxygenic and then talk about anoxygenic phototrophic organisms. Oxygenic, अगर तुम्हें याद हो तो ये higher organisms या cyanobacteria में होता था. Now, here in oxygenic, you have two distinct sets of reactions. Okay, for us, the prime concern is the light reaction, क्योंकि तुम यहाँ देख रहे हो ATP generate हो रहा है, NADPH generate हो रहा है. Dark reaction is when CO2 is fixed. Now, while you will come across these two terminologies, light reaction and dark reaction, I have to mention to you that this is a misnomer. So earlier it was believed that it is only this set of reaction which requires light, and dark reactions are independent of light. But right from this figure, you can understand the dark reactions will not proceed unless there is NADPH, the reducing power, and the ATP which funnels or fuels the dark reaction. So अब ये लाइट इंडिपेंडेंट कहते हैं कुछ लोग, या मतलब डार्क रिएक्शंस बुक्स में अगर देखो तो भी समझ जाना कि दिस इज अ मिसनोमर. सो मोस्टली वी आर कंसंट्रेटिंग ऑन द लाइट रिएक्शन. ओके, now you have to really understand the students, huh? Like I mentioned, the cyclic and the non-cyclic phosphorylation processes. Here what happens is, in the case of oxygenic, you have two photosystems. A photosystem kya hai? This entire complex of the main pigment as well as the accessory pigments. Now what happens here is, there is a photosystem 1 which is called as P700. Actually 700 is the wavelength of the absorption maxima for this chlorophyll. So what happens here is, once photon hits this reaction center, this passes on its electron to the electron transport chain, but here it actually goes to NADPH. तो हम लोग ने नॉन साइक्लिक फोटोफॉस्फोरिलेशन की बात की थी. If it is being distracted, it's not coming back to P700. You need something to actually replenish the electrons which have passed on from photosystem 1. And in the case of oxygenic, there is another photosystem. Now you have to look at the difference in the electron energy. Okay, the redox potential. This is lower. P680 is lower. Now what happens is, there's another reaction center which is termed as P680. P680 again in a similar process, the photons strike and there is electrons which move on to the first electron acceptor. It passes on through an electron transport chain. Now whenever I say there's an electron transport chain, you have to understand that there is a proton motive force being generated and ATP is thereby synthesized. The purpose of this photosystem is ATP generation as well as replenishing the electrons which P700 has lost. ठीक है? So this replenishes P700 and P700, अब क्यों ये हो रहा है, ये तुम लोग शायद ये पूरा देख लोगे तो शायद तुम्हें समझ में आ जाए. Why do we even need two photosystems? तो P700 का purpose है formation of NADPH, while P680 का purpose है ATP generation as well as replenishment of the electrons to P700. And in certain cases, actually, this scheme is called a Z scheme, students, okay, because of the kind of the structure that you see here. Under certain conditions, what happens is, the photoexcited electrons from P700 takes an alternate path. It can actually pass on to, from the primary acceptor, it can pass on to the cytochrome complex such that it returns back to 700, only in certain cases. Okay. And these cases include, for example, if the cell, if the cell suppose has enough of NADPH, it has enough of reducing power, it can keep generating ATP by not letting the electrons pass on to NAD+, instead moving it to cytochrome complex. Students, समझ में आ रहा है ये जो हम बोल रहे हैं? कोई एक मुझे कंफर्म करेगा? जस्कीन?
Yes, ma'am.
साइक्लिक नॉन साइक्लिक समझ में आया था ना? इसीलिए मैंने पहले साइक्लिक नॉन साइक्लिक बताया, उसके बाद हम तुम्हें ऑक्सीजनिक फोटोट्रॉप्स बता रहे हैं. जयेश, कुछ बोलना चाहते हो?
नो मैम, समझ में आ गया.
समझ में आ गया, ठीक है. तो अब तुम्हें समझ में आ रहा है ये चीज कि क्यों हमें दो फोटो सिस्टम्स चाहिए? एनीबडी? कशिश, can you tell me why there is a requirement of two photo systems, Kashish?
Ma'am, actually, I have not even covered the previous videos, so this video is not exactly clear. So I'm not sure exactly.
अच्छा, anybody else? बताओ. दीपक बता सकते हो, मैसेज कर सकते हो. ध्वनि? हाँ, आशी, tell me.
Your one photosystem will be responsible for synthesizing NADPH, whereas that is P700, and the second P680 will be synthesizing both P700 as well as NADPH.
अच्छा, अब ये बताओ कि why can't P680 be the only one responsible for ATP generation and NADPH generation? बोलो. Why can't one photosystem function in both?
Ma'am, is it because of the cytochrome complex?
It is because of the redox potential actually. P680 इसका जो primary electron acceptor है ना, it cannot give its electron to NADP plus. So NADPH cannot be generated by P680. On the other hand, P700 का जो primary electron acceptor है, it is in a position to actually donate electrons to NADP plus for formation of NADPH. So it's 700 that can give it, huh? लेकिन P680, so the photolysis of water which donates electron to P680 will not be able to do it at P700. So you actually need another complex. Water cannot directly replenish P700 ka electron. So it has to give it at a reaction center which is lower in redox potential than P700. Okay, so it's all about the redox tower. Okay, Ashi. So one
Yes, ma'am.
photosystem really will not be sufficient here. Okay. ठीक है. So again, this I'm not going to go into the detail. ये वो था it's light harvesting and ATP generation process, but here carbon dioxide exactly the same thing. Calvin-Benson cycle होता है and वही reduction पहले carbon fixation. Rubisco is the enzyme. We'll not cover this. Coming to anoxygenic photosynthesis, इसमें देखो क्या होता है. Anoxygenic and oxygenic are different in the sense that there were two photosystems, but in anoxygenic phototrophs there is only one photosystem. Okay, for the conversion of light energy to chemical energy. Here, as you can see, there are two organisms or two types of organisms that I am looking at here: purple bacteria and green sulfur bacteria. Again, a range of intermediates are involved in this transfer. Look at the difference in the redox here. One is a P870, the other one is a P840. Again, these are the absorption maxima that we talk of. Now, इसमें एक चीज देखना, while in the case of green sulfur, the electron acceptor can actually transfer its electron very easily to form NADH. In the case of purple bacteria, it is the similar case that you saw in the case of chemolithotrophs, wherein for the formation of NADH, reverse electron flow is required. ये देखो, ये जो ये movement दिखा रहा है, that is talking about this having a higher negative redox potential compared to Q, which is quinone, a central lipid carrier जो तुमने chemolithotroph में भी देखा था. So again, for purple bacteria, they need to invest energy to synthesize NADH. Okay. Now, if given a choice, if a purple bacteria is given a choice between photoautotrophy and photoheterotrophy, you can understand that it would not like to enter into this mode wherein ATP is diverted to synthesize NADH. Okay. So it all depends on what kind of sources are there. This is an energetically unfavorable process. But the green bacteria, they can, of course, they can always prefer to go into the autotrophic mode by alternating this use. So here that is not an issue. I mean, reverse electron flow is not required here. But for purple, of course, they would exist as photoheterotrophs, so that there's maximum optimum utilization of energy. And this happens to be the last topic, which is kind of different from what we saw till now. So oxygenic तुमने देखा, anoxygenic तुमने cyclic होता है, oxygenic में cyclic and non-cyclic होता है. I hope you understood when I said कि यहाँ से ये cyclic भी हो सकता है. तो cyclic अगर होगा तो तुम समझ रहे हो कि NADPH जनरेट नहीं होगा. It will only happen in the case wherein NADPH is in sufficient amount in oxygenic photosynthesis. यहाँ पे it's only cyclic, right? Now in archaea, there is a separate kind of phototrophy that takes place. Here there are rhodopsin-like proteins which absorb light energy, and they do not transfer electrons, or they are not associated with any light antenna. In fact, the special thing here is that it is the pigments, that is bacteriorhodopsin, which is the one which is harvesting light as well as serving as a proton pump. जो तुमने अभी तक नहीं देखा था. You always saw that the right harvesting complex, whether it was oxidative phosphorylation or photophosphorylation, there was an electron transport chain involved, which led to the efflux of protons, and then that proton motive force was used in ATP synthesis. However, here it is the pigment itself which is functioning in this pushing out of proton, okay, and generating the PMF, right? So this is quite unusual, but these rhodopsins, they are found in, they should actually be called archaeorhodopsin, but they're called as bacteriorhodopsin. They're only found in archaea, okay. So it undergoes a conformational change, pumping protons across the cell membrane, and thereby there is no electron transport chain involved in rhodopsin-based phototrophy. बस इतना कि ऑक्सीजनिक में there was only the Calvin-Benson cycle which was involved in carbon fixation, but in anoxygenic photosynthesis you have Calvin-Benson as well as two other carbon dioxide. Yeah, so this is just one link that I thought would be was interesting. Now, students, I'm open to question. Tell me if you have something, any doubt here. I know that we've been covering two pathways, two nutritional types today, so there might be questions you can ask. कुछ doubt है chemolithotrophy? दो तीन लोग ही बचे हो तुम लोग, you can tell me.
Nothing as of yet, because I guess I'll have to